Hello, everybody. Welcome back to another episode of the Lil Dudes Insect Academy podcast. Wait, before we get started, I just wanted to say thank you for tuning into this episode of the Lil Dudes Insect Academy podcast, and just ask you to please hit the like button below, or click the subscribe button if you're watching on YouTube, or follow this podcast if you are listening. That really helps the show out. But also, if you want to help us further, then join our Patreon. We have an awesome community that is growing there. Or if you want to make a one-time donation, we are a nonprofit foundation, so head to our website. Both the Patreon and the website will be linked down below. But without further ado, back to the episode. I'm your host, Bradon Coy, and I'm excited to be here with Dr. Jordyn Barr. Jordyn and I met at ESA back in November, and she is a graduate student at Vanderbilt University, and she's done a lot of work with mosquitoes, which we've talked about a lot on the show in the past, but I'm excited to hear about her exciting research and stuff that she's done with them. And so, without further ado, welcome to the show, Jordyn. Go ahead and introduce yourself for us. Thank you so much for chatting with me.
Hello, thank you so much for having me. So I'm Jordyn. I am originally from just outside of Erie, Pennsylvania. I then went to Ohio Northern University for my undergrad and got my degree in molecular biology. And I really was inspired to pursue research, and I did a lot of work with Drosophila genetics, and I have additional research experiences with them. And then after my undergraduate degree, I pursued my PhD at Vanderbilt, started in 2019, and I joined an interdisciplinary graduate program. So I came in not really knowing what I wanted to do, other than that I loved research. And so I rotated through a series of different labs, ended up joining Julian Hillier's lab here at Vandy, where I work on mosquito physiology. And my project over the last 6 years has really been focused on this interplay between how two different factors affect mosquitoes' ability to survive and respond to infection, looking at environmental temperature and the age of the insect. And then I recently defended 3 weeks ago, and so I'm just finishing up my PhD here at Vanderbilt before I go on to pursue my postdoc.
Congratulations on that. That's
Thank you.
Super exciting, and we're going to talk about all of your awesome research. So you are freshly Dr. Barr. That's very exciting.
Exactly.
All right, so I guess we'll just start from the beginning and sort of walk through your research career so far. Sort of dive in for us a little deeper on, starting with that Drosophila research you started at first. Sort of break that down for us. Like, what were some of the things that you learned there and the interesting things you may have found? And for those listening, Drosophila is the fruit fly, the quintessential model organism that we use for just about everything. But yeah, tell us a little bit about that. How was that for you?
Yeah, so when I was originally looking at where I wanted to pursue my undergraduate work, I chose Ohio Northern, and I was between pursuing something with environmental science or pursuing a more molecular-based research and degree. And my mentor at Ohio Northern, Dr. Jamie Siders, really inspired me to pursue molecular biology. She runs a fruit fly genetics lab, and during the spring of my freshman year, she pulled me aside and sat down with me and was like, hey, I have this really great research opportunity in the summer to get you started in the lab early, and think that you would be a really great fit. So my first introduction really into a research lab was the summer after my freshman year, where I learned about fruit fly genetics and learned about a specific protein called CASC that's involved in this group cell movement. And so my work in her lab then for the next 3 years really focused on the role that this protein plays in group cell movement during ovary development. And so as egg chambers develop in fruit flies, there is a cluster of cells that migrates from one end of the egg chamber to the other to form this micropile for sperm entry so the egg can be fertilized. And this group cell movement is really a model for group cell movement in wound healing. And so the main question we focused on is, what is this protein CASC? What is its role in this group cell movement? Is it guiding the cells? Is it part of the cell cluster? Is it using like a signaling molecule? And so my role in the lab really was to make these transgenic fly lines to label this protein with green fluorescent protein so that we could visualize it as these group cells move from one end of the egg chamber to the other.
Wow, yeah. So when you say like group cell movement, is that like when the fruit fly becomes injured, or like what do you mean by that?
Yeah, so it is a model for wound healing, but in the fly itself, we're looking at it during a developing egg chamber. We're thinking about like before there's actually an egg in the female fruit fly. It goes through about 14 different stages of development. And there's a cluster of about 10 cells that kind of forms this ball structure that kind of tumbles from one end of the egg chamber to the other. And so we were really interested in understanding how it's able to kind of maintain polarity, how it's able to know what direction it's moving from one side of the chamber to the other, what is guiding it, and everything like that. And so this protein cask is a scaffolding protein in other systems and is highly expressed in this system. And so we were really wondering what its role was in guiding this tumbling group of cells that's moving while the egg chamber develops.
So, like, did you find anything interesting? Like, what did you guys discover there?
Yeah, so like a lot of undergraduate research projects, research kind of moves a little slowly when it's only led by undergrads in the lab. So Ohio Northern does not have any graduate students in the biology program. And so it's a really awesome opportunity for undergrads to kind of step up and lead and guide the research questions. But it also means that research doesn't move as quickly as it does when there's graduate students in the lab to continue the research when an undergrad is not there. Nothing really came out of my work there other than I got a lot of lab experience. I produced these different fly lines. Those fly lines were able to be used for future years. I'm not sure that if they were used. We took some really awesome pictures of this group cell movement, and I got a lot of really good experience mentoring other undergraduate students and learning how to make poster presentations and share the beginnings of this research with people. So it was a really great opportunity to learn some skills in the lab, learn how like projects begin, but unfortunately I didn't see it to an end.
Okay. Yeah. What a cool opportunity though. Like for those of you listening, like getting especially 3 years of undergraduate research experience is pretty rare. Like, because there's always, the graduate students are obviously the priority. They're what the school is paying for. Undergraduate students are kind of more of a volunteer, like elective kind of thing. Like if an undergraduate student wants research experience, they would talk to the professor or whatever. But
yeah, exactly. So this whole thing was
rare.
all volunteer, and because I was the main person on this project, it's like I was the one troubleshooting, working with the PI, helping to order supplies, things like that, which is a unique experience for an undergrad when you're putting in volunteer hours during the week as opposed to trying to spend all of your time in the lab.
That's so cool. Okay, so next up, you did grad school next. So tell us a little bit about that. Like, what was that? What was that project and how did that go? And it sounds like you had a lot of experience from Ohio Northern that really helped you be a lot more prepared for that, because you've already basically been a grad student, I mean, functionally. So, like, you've already ordered lab supplies and all of those things before. So it sounds like you were, like, really well prepared for the next step. So tell us a little bit about that.
Yeah, so I guess also a little bit before that. So Ohio Northern's a small liberal arts school in middle of nowhere, Ohio. My first summer after college, I worked at Ohio Northern getting research experience in the lab. My other summers, I was able to work in other labs, such as at the Scripps Research Institute down in Florida or at Columbia University, and I got additional research experience from those larger institutions. So those were 10 or 12-week summer internships where I was working on a small project with a graduate student, kind of seeing those things carried to fruition.
That's awesome.
Experiences really gave me the opportunity to work alongside graduate students and postdocs, work at larger institutions, see what research at an R1 institution really looks like, talk to graduate students to see if graduate school was the right path for me, and see different experiences of professors at these different institutions. And so I felt really confident in when I went to apply for graduate school that I knew what I was looking for and that it was the right career path for me. Knew that I wanted to stay in academia, academia, or at least that's what I thought when I was applying for graduate school. And I knew that I wanted to broaden my research experience. So each of those experiences, I continued to work with fruit flies, asking different questions and using them as a genetic model system to answer additional questions that could be applied to human systems. And so I knew that I wanted to broaden my experience. Maybe I didn't want to work with fruit flies forever, even though they're an awesome genetic tool. And so when I was looking at graduate schools, I was looking at umbrella programs, or basically programs that allowed me to apply to one program, but I could rotate in labs from across a variety of different departments so that I could experience rotating in labs that are asking different questions, that are using different techniques, that are in different programs that provide opportunities for teaching or for internships or for different presentations depending on the different collaborations that are going on in those different programs. And so that was really what I was looking for. I applied to graduate schools kind of within a specific region, and I had 7 interviews and 7 offers. I was like,
wow.
Man, I have
lots of choices.
I have, all these options, and so I did feel very prepared going into those graduate school interviews and everything like that with my research experience. Vanderbilt was the last of my interviews. Clearly they stood out because it's where I ended up coming. Mm-hmm. And I specifically came for the IGP program, or the Integrated Graduate Program. And so this is their version of an umbrella program that encompasses about 12 different departments on campus.
Oh, wow.
And so I chose Vanderbilt because I felt like they really supported graduate students. They had career support through something called a Brett office, which basically offers different internships, like CV writing skills. They offer different workshops to help cater your skills for interviewing, for applying for faculty positions. They have these talks called failure talks that basically bring in professionals in the world, and they tell you the mistakes that they made, and they're still doing great things, great science. Mm-hmm. They have a center for teaching, so I felt like I could be supported in that realm. And importantly, I could choose across 12 different departments kind of where I wanted to rotate. And I had the opportunity to then rotate in 4 different labs before making my selection. And I felt like that gave me a lot of opportunity to explore different questions in science to really broaden my research before kind of selecting what I wanted to do for graduate school.
Yeah. So at Vanderbilt, you just finished up mosquito research.
I did.
How did you like, you said, I think 12, did you say 12 different options of like
12 different departments, yeah.
Yeah. So like, how did you finally, settle in on mosquitoes? Like, what about them like brought you into that?
Yeah. So So Vanderbilt does not have specifically an entomology department. I don't have an experience in an entomology department. I came from a molecular biology department, a small biology background. So there wasn't specifically like an entomology department at Vanderbilt that I was looking in. Really, I was picking labs where I was interested in the question, but more so that it would give me an opportunity to work with different systems. So I came in having fruit fly research experience. I rotated then in a C. elegans lab, which is a small worm nematode that's also used for a lot of genetic screens, genetic systems. I then rotated in the mosquito lab, which my PI likes to remind me that I'm no longer using a genetic system. I'm using the system. So he likes to say that it's a step up. Then I rotated in a cell culture lab looking at coronaviruses, which
interesting,
ironically was right as the COVID outbreak was happening.
Really? Wow.
I rotated there January through end of February of 2020. And then I rotated briefly in a yeast genetics lab. So again, really broad research interests, broad research questions, but the goal was to gain different skills and different tool sets that I could then apply to any question that I decided to pursue next, which is kind of a different strategy than some people come into graduate school with. Some people come in with a very specific set of questions that they are interested in researching, and they rotate in a whole set of labs that ask questions that are similar, and they're like, I am joining a research lab that will focus on diabetes research or something like that. And you're like, okay, that is much more narrow of research interest than what I had. So I rotated through all those labs. I really enjoyed a couple of my rotations, but things that stood out about the lab that I ended up joining: the mentorship style, the lab environment that I ended up joining, the fact that there was a brand new grant that was getting started and I could kind of help dictate how that grant was carried out.
That's cool.
So it gave me a lot of opportunity to kind of be a leader early on in the lab,
Mm-hmm.
as well as the department itself that I ended up joining. So I ended up joining just the biological sciences department at Vanderbilt, which is kind of their general biology department. But it's the only department that's not, or basic sciences department that's not affiliated with the med school. Instead, it's affiliated with the College of Arts and Sciences, which
Cool.
means that all of the faculty have teaching appointments. And so we are directly, we are required to be a teaching assistant for at least one semester. We directly work with undergraduate students. They come to work in the labs in this department. So it gave a lot of opportunity for mentorship and teaching experience, which is something that I really wanted to gain in graduate school. And so I felt also very supported in that type of professional development experience. And so that was another big pull to join the lab.
What a cool opportunity, though, because we have a similar system at UF, sort of. Well, I mean, I guess this is kind of a thing across universities where you have this big school, right? But then you have all of these different colleges within the university, and then even within the colleges, you have different departments, right? So, for example, I'm in the entomology department. We're within the College of Agricultural and Life Sciences, which is this huge college at the university, but then the department is super tiny. So you get the benefits of the resources from the huge college, but then the personal nature and like intimacy of the tiny department, right? So
Exactly.
it sounds like you got a similar experience at Vanderbilt, which is really awesome. So
Yeah.
I'd love to hear that.
Yeah, exactly. Yeah, definitely. I would say that we are a tight-knit department we kind of overlap between the departments that are in this basic sciences affiliated with the medical school. So we have all the resources of the, medical school research cores and career centers and the collaborations that can be across labs and things like that. But we are also unique in that teaching experience and mentorship experience that we can gain with the undergrads.
So would you say that you're more of like a general biologist, or like, are you super into science in general, or like, do you know a lot about bugs, or like, where do you sort of fall on that? Because we met at the entomology conference, the meeting. So where do you fall on that range of bugs versus science
in general? Yeah. So at this point, right, I just defended I have my PhD from a mosquito physiology lab. I worked with fruit flies. You're thinking to yourself, surely
those are both
sort
bugs.
of a bug person, right? Ironically, though, I really consider myself sort of a fake entomologist. I've never taken an entomology course. I never saw myself pursuing something like a degree in entomology or a career down this path. I think my family would say, you hate bugs. Why, why are you pursuing that? But I have always really enjoyed I've enjoyed scientific investigation, curiosity. If you remember, I talked about in my decision for an undergraduate institution, I was between molecular biology and sort of an environmental science ecology route. And so I've always been drawn to something in that direction. And I found that my work in the lab that I'm in now kind of balances that, where we do a lot of basic science research looking at the biology of the insect, but applying different environmental outcomes, specifically with climate change and warmer environmental temperatures and what that then does to the physiology of the insect. So I would say that I am not the largest bug enthusiast, but I am happy to continue researching them.
Right, yeah.
And I would say that I can get excited about a lot of different questions in science. And I find myself being involved in discussions with other graduate students, with my friends from other departments and things like that. And I'm like, wow, your work is totally different on a day-to-day than what my work is. But I can still appreciate it and get excited, and could have easily seen myself pursuing a different research topic as well.
Absolutely. And for those listening at home, I just want to emphasize, you can do so many things with bugs, and you don't have to get your degree in entomology, because honestly a lot of schools don't have an entomology program, so you're not really limited at all. So you can get your bachelor's degree in biology or really anything, and then you can move on to work with bugs later, and they apply to everything, and especially mosquitoes. There's a ton of research happening with mosquitoes for a lot of reasons.
Yes.
So you can really go anywhere with bugs, even if you're not like the stereotypical bug kid as I am.
Exactly.
So, yeah. And they apply to everything with science and ecology and human health and all of that stuff. So it's really, really awesome. So I want to talk about your mosquito research a little bit more. Like, what specifically did you just finish up defending? Like, what were some of the interesting things you discovered along the way, and like, how was that whole experience for you?
Yeah, so my project in the lab is lovingly termed 1/2 of the temperature grant. What does that really mean? It means that I really looked at this intersection of how warmer environmental temperature altered the aging process in mosquitoes. And so with warmer temperature, we're seeing a rise in body temperatures of insects as well. So insects are poikilotherms and ectotherms. And what that means is that they get their heat from their environment and they do not use their metabolism to thermoregulate or to actually maintain a stable temperature in their body temperature. And so with that being said, with climate change, mosquitoes then are experiencing warmer temperatures than they become accustomed to. And with that rise in temperature, their body temperature is also higher. So because body temperature then dictates the rate that a number of different physiological processes happen in the mosquito, we're seeing faster rates of development, faster metabolic rates, and changes in the way that they're able to respond to infection. And so this is really important because climate change does not affect every region the same way. Some regions we see a larger rise in temperature than others. And so we're seeing changes in the geographic distribution of the mosquitoes and other insects as well.
For sure, yeah.
And so more people may be exposed to mosquito-transmitted diseases. And so it's really important to understand really what is happening with these warmer temperatures on the biology of the mosquito itself. On the other half of that, the other factor that I was really interested in was the age of the insect. So as mosquitoes age, they undergo a process called senescence, which is characterized by an overall deterioration of their body condition, weakening of their immune response, and a slowing of their metabolism. With that, we see an increased rate of mortality, and we see a reduction in their ability to reproduce. And senescence is really something that all living organisms undergo as they get older. So, as you and I age, we are also experiencing these different deteriorations of our body condition. And this aging process can be characterized in two ways. So we characterize it by their chronological age, or how long an organism has been alive. And we also characterize it by an organism's physiological age, or how well their body is able to function. In homeotherms, or mammals like you and I that use our metabolism to thermoregulate, those two processes typically match or are coupled. So however long we've been alive typically corresponds to also the way that our body is able to function. However, in poikilotherms, like mosquitoes who do not use their metabolism to thermoregulate, their aging process may be decoupled, and they may be aging faster or slower physiologically with changes in temperature. Where we see slowing of different processes in cooler temperature, and we see an acceleration of different processes in warmer temperature. And so my work really focused in on what was happening with climate change with these warmer temperatures, looking at the question, does warmer environmental temperature accelerate the rate that mosquitoes age? And then thinking about kind of what that has as far as outcomes for disease transmission dynamics. And so for the first set of questions I looked at is how their body size and body condition may change with warmer temperature and aging. And we use this kind of as a baseline to determine the quality of the mosquitoes, right? How long are they able to live? Are they likely to transmit disease? Are they strong enough to reproduce? Things like that. And so for my experiments, I reared mosquitoes at 3 different constant temperatures, 27 degrees, 30 degrees, and 32 degrees Celsius, which are really hot, right? So we're simulating a warming environment. We're staying within a range that the mosquitoes will actually survive, right? We're not trying to just immediately kill them, because if we are just cooking them, they're not going to transmit disease. And then I'm monitoring different aspects of their physiology across their entire lifespan, but really measuring them at 4 different ages, 1, 5, 10, and 15 days into adulthood. So we can really capture the effects that occur immediately after they eclose as adults, and also much later into the aging process, so we can really see how their body changes over the course of their lifetime. with those different temperatures. And so with that first set of experiments, I saw that mosquitoes were just slightly smaller with warmer temperature, which was kind of to be expected, but they did not change that much in their overall body size. So we could kind of rule that out as a confounding factor for later on measures of kind of strength of the mosquito. And something that we really focused in on was that interaction between temperature and age. Does temperature alter the rate that they age? Changes in their carbohydrate content, their lipid content or their fat content, as well as their protein content as just a measure of their body composition. And we saw evidence of this interaction between temperature and age with their protein content, where we saw their protein content decreased with warmer temperature and decreased with aging, and this aging dependent decrease occurred faster and occurred earlier in life when the temperature was warmer, suggesting that these mosquitoes are using these proteins as they age at a faster rate when the temperature is warmer. So we see evidence of this interaction suggesting they're aging faster. But what does that really mean now if they're, if we're seeing a change or deterioration in their body condition at a faster rate? What does that mean with their ability to survive? So I next measured changes in their survival, and I measured these not only in just naive mosquitoes, but also when they were immune challenged, because we want to know how a mosquito is able to survive if they are carrying some sort of pathogen that can then be transmitted to humans.
Yeah.
Right? And so I saw that, survival decreased in the presence of infection with warmer temperatures, and as they got older, they were not able to live as long. But we saw evidence again of this interaction where warmer temperature accelerated or sped up that aging-dependent decline in survival, where mosquitoes that are younger at these warmer temperatures had similar survival outcomes to mosquitoes that were older at these cooler temperatures. Not able to live as long, potentially because they're deteriorating with their protein content faster, but also there's other factors that are contributing to that. And we really saw an acceleration of this when it was in the presence of infection. And so we wanted to know then, is there a change in their immune response? And so the first set of investigations looked at changes in this intensity of a bacterial infection as an indirect measure of their overall immune function. And so I was able to measure changes in the intensity of an infection with greater intensity of an infection correlating to a weaker overall immune function because they're not able to control that infection as well. And then vice versa with a lower intensity of infection correlating to greater immune function. And so through a couple sets of experiments, I showed that warmer temperature infection intensity, so the infection is much greater at warmer temperatures. With aging, we see an increase in infection intensity, so older mosquitoes are not able to control that infection as well. And we see an acceleration of that aging-dependent increase in infection intensity with the warmer temperature, meaning that younger mosquitoes at a warmer temperature are not able to effectively control that infection, as well as older mosquitoes at a cooler temperature. So we're seeing this greater intensity of an infection earlier in life, makes you question, are mosquitoes more infectious than at warmer temperatures? Even if they're not surviving as long, can they be more infectious?
Yeah.
And again, that set of experiments was an indirect measure of their changes in immune function. And so we really wanted to know what specific immune processes are being affected by warmer temperature, aging, and their interaction. And so another graduate student in the lab, Lindsay Martin, has really focused on one half of that set of immune function, whereas I've focused on another half. So mosquito immune systems are made up of both cellular and humoral immune components. Cellular immunity is really mediated by hemocytes, which are the main immune cells in the mosquitoes, and their main function is to phagocytose pathogens, or eat and sequester the pathogen itself to control the infection. They also then produce other immune effectors that are secreted into the hemolymph, or the blood or fluid that is circulating through the insect's body. And the immune processes that are then that occur in the hemolymph are part of the humoral immune function. And so these can be different antimicrobial peptides that perform lysis, or something called a melanization cascade, which deposits melanin on pathogens to sequester the infection. So Lindsay focused in on the humoral immune response, and she found evidence that warmer temperature accelerated this aging-dependent weakening of immune function for both the melanization and the lytic responses.
Interesting.
So, both of those processes weaken with aging. That's a that is part of senescence, a characteristic of senescence. But we're seeing it occur faster at these warmer temperatures. And so my work then wanted to balance that and investigate if we saw similar trends on the cellular immune response, right, or this hemocyte-mediated response, or if the hemocytes were actually maybe increasing their function to compensate for the weakening that we were seeing for the humoral immune function, right? And so to measure the strength of the cellular immune response, I quantified the number of hemocytes in 2 different spatial locations. So hemocytes can be found circulating through the hemolymph or flowing through the body cavity, pumped through by the heart, and those are called circulating hemocytes. And they can also be found attached to different tissues throughout the body cavity, and those are called sessile hemocytes. And circulating and sessile hemocytes can change locations. So circulating hemocytes can attach to tissues, and those that are sessile can detach and enter circulation. And those 2 spatial classes work together to control an infection throughout the body cavity. So I quantified the number of hemocytes, and I also measured changes in their phagocytic activity. So phagocytosis is their main function, and so I quantified the number of cells that were performing phagocytosis, as well as the number of bacteria that those cells that are performing phagocytosis actually were able to sequester and capture within them. So we wanted to know not only the number of cells available to respond to infection, but how active those cells are. Because just having a bunch of people in an army to respond is not the same as actually having a prepared immune army to attack and control an infection. And so we found evidence that for circulating hemocytes, we saw a decrease with warmer temperature, a decrease in the number of cells with aging, and a faster aging-dependent decrease when the temperature was warmer. So again, we saw this accelerated weakening of the response. And for the sessile hemocytes, we saw evidence of this also in the presence of an infection, where we saw that aging-dependent decrease occur earlier in life when the temperature was warmer. So we're seeing less immune cells earlier in life. Mosquitoes have a weakened, faster weakening of their cellular immune response. We're looking then at the activity of those immune cells, and we see that the activity increases with aging. And so perhaps then the fewer immune cells that are available to actually respond to infection have to do more in the mosquito to actually control that infection. So they're taking on a greater phagocytic burden. And so we're seeing an increase in the number of immune cells responding or actively performing phagocytosis, an increase in the capacity of those cells, and we're seeing an acceleration of that aging-dependent increase with the warmer temperatures. So overall, we're seeing that warmer temperature is accelerating cellular immune senescence in the mosquito, just like it's accelerating humoral immune senescence. Basically, my work shows that mosquitoes are aging faster when the temperature is warmer. But we not only have to think about the effect that this has on mosquitoes. So mosquitoes are poikilotherms and ectotherms, but most insects are too. So insects that are, pollinators, insects that are agricultural pests. This means that they are likely aging faster as well when they're exposed to these warmer temperatures. And so really, what does that mean for pollinator success? And what does that mean to our ability to actually control agricultural pests in addition to our disease vectors? Is their physiology responding the same way to different insecticides, or are they having new ways to actually evade those mechanisms with a change in the rate that they're aging? So, it brings up different things to really think about outside of just insect vector systems, disease vector systems. We need to think about what is happening with the effects of warmer temperature more broadly. And it's really exciting to show, that we can change the rate that insects are aging and kind of think about what other questions that we are able to ask in those senses. And so the lab is continuing to work on, looking at changes in reproduction, looking at changes in fertility and fecundity. And also we're looking at, we've looked at all these phenotypic changes and these phenotypic outcomes of the mosquito. But what is happening at the genetic level? Are we seeing changes in the gene transcription profile? What is the mechanism behind this accelerated aging? And so that's kind of the direction that the lab is going currently, and it's really exciting to see, the other questions that have really stemmed up from the project.
Yeah, I mean, there's so many implications that I have been thinking about as you were talking, and that was actually one of my questions, like, is there a genetic aspect to it? Because climate change is pretty slow in comparison to the lifespan of an insect.
Right.
actually very slow, right? Climate change in general is very gradual and slow, and so could we be seeing a change in the genetic expression of certain genes through insect development? Is a pretty interesting thing to think about, and it sounds like they're trying to answer that question now from what you were saying.
Yeah, so we're really excited about an RNA-seq project that's coming out of the lab that really looks at these gene expression profiles from each of our different mosquito populations reared at our 3 temperatures at our 4 different ages to kind of see how changes in gene expression occur, within the lifespan of a mosquito. What you're thinking about is really what is happening sort of across populations and considering evolutionary effects and things like that. Unfortunately, the way that our experiments are set up, we are not able to track, this specific population from egg to adulthood, and then those adults laid eggs, and we're continuing, 6 generations at those warmer temperatures to see what would actually happen. Surprisingly, we are really at the extreme temperature for our lab-reared mosquitoes, and they do not like that 32 degrees constant temperature. And so we see a significant decrease in the number of eggs that they're able to lay. And so we are not able to maintain a population just from that temperature. And so that in itself really tells us that, these warmer temperatures are causing some significant changes in the mosquito.
So if you're not able to, just like a curiosity, if you're not able to, maintain a reliable colony at that temperature, do you typically raise them at a lower temperature and then introduce them at adulthood to that higher temperature, or how does that work out?
No, so we have colonies of mosquitoes that are at 27 degrees. Those colonies are the ones that are blood fed, and we collect those eggs, and then the eggs are distributed between each of our 3 different temperatures. And those eggs then are reared to adulthood, and those are experimental mosquitoes. So those mosquitoes experienced those warmer temperatures from egg to adulthood to time of experimentation. So they are experiencing the effects of those warmer temperatures during all the larval developmental stages as well as into adulthood. It's just the parents themselves that are laying the eggs. They're not necessarily from the exact same temperature.
Okay. So what you're saying is the experimental population, once raised to adulthood, don't really like to reproduce as much as the lower temperature. Got it. Okay.
Yeah.
Yeah, there's so many implications. There's the broad one that you were talking about with insects as a whole, right? Insects as a whole will all be affected by this senescence thing you were talking about, right? They all age in some respect, and with temperature, it's a really interesting thing to think about. But then also, there's the disease transmission aspect of it, which sounds like to me the pathogens actually are transmitted better at higher temperature. Did I understand that correctly? Well,
they could be, right? So we measure disease transmission potential using something called vectorial capacity, which basically is a measure of the potential human infections that could come from one infectious mosquito. And it accounts for a variety of different factors of mosquito physiology, pathogen physiology, and it also accounts for factors that are part of the system, outside of the system, and interactions of those factors and how they impact the different parameters. And so my findings really contribute to 2 different aspects of vectorial capacity, one being mosquito survival. So we saw a significant reduction in mosquito survival with warmer temperatures and at older ages. And so with a reduction in survival, you would predict a reduction in the ability of the mosquito to actually transmit disease because they are not able to live as long, and they may not survive the period of time needed for the pathogen to mature within the mosquito before the disease can then be transmitted. However, we also saw a weakening of their immune function overall, right? And so that directly relates to something called vector competence, or the ability of the mosquito itself to not only support pathogen development, but also combat pathogen development. And so immune function is just one factor or one component of that. And so if we see a weakening of their immune function, mosquitoes may be seen as more infectious, like I mentioned, with greater intensity of infection. Perhaps that means they're more likely to transmit disease. But on the other hand, if they're experiencing a greater pathogen load, a greater intensity of an infection, they again may not actually be able to survive that infection. And so that may in turn be contributing to that decrease in survival. And so if they're not able to survive long enough to actually transmit disease, would we see an increase in disease transmission? That's really what we're trying to understand. There's a lot of other different questions that then go into like pathogen development. So all of my work has really been on the biology of the mosquito. But any factor that we're investigating in the mosquito is also in turn impacting the development of the pathogen within the mosquito itself.
Right.
So with warmer temperatures, pathogens are developing faster within the mosquito. They don't need as long to live or to get to a stage to actually be transmitted. And so do mosquitoes actually need to live as long to transmit disease as we were originally predicting? So I keep talking about this like period of time of pathogen development. So vectorial capacity takes into account like a mosquito biting rate and this incubation period for the pathogen. And those two things kind of together really, you know, a mosquito needs 2 bites, 1 bite to pick up the pathogen, the next bite to transmit the pathogen. That time in between those bites can range in the time that it takes for a pathogen to not only enter the mosquito, exit the midgut, and then migrate back up to the salivary glands to then be transmitted. And so if they're developing faster, the time between those 2 bites decreases, and the mosquito may actually, you know, a decrease in survival may not actually change the mosquito's ability to then transmit disease.
That's interesting to think about because like you said, maybe the time between those 2 bites might be shorter, but also the development time of the pathogen might be shorter as well. So it might just balance out to where it's not changed at all.
Right. Yeah.
It's so hard to like simulate in the lab.
It's really
I'm sure.
hard to simulate in the lab, and we have to remember that, you know, what we're doing is not the natural pathogen a host system, right? We're using bacteria, which is it simulates what a systemic infection would be. But the mosquito system that I use are Anopheles gambiae, which are the African malaria mosquito. And so they are the main vector for Plasmodium falciparum, which is the pathogen that causes malaria in humans in tropical Africa. And so, you know, we're not exactly measuring that relationship between the exact host pathogen species and what that would look like. Right. What we are able to measure is kind of what happens once that pathogen enters the hemocoel and it becomes a systemic infection, and that's what we are able to simulate.
So interesting to think about. Yeah.
And like, you know, there's thousands of species of mosquitoes. They transmit, you know, hundreds of different pathogens. Each of those pairings has a unique, you know, vectorial capacity prediction. And some of those mosquito species are much hardier than ours. And so slight changes in temperature may not have as drastic effects. So just because they might already be naturally found in areas where the temperature varies much more extreme, or they may have evolved different adaptations to kind of counteract those extreme temperatures.
Wow. This is, I mean, so interesting. And, like, I've had a couple other mosquito researchers on, but, like, everyone has such a different, like, field that they're working on within mosquitoes. I mean, there's so many different things to look at, and they affect us in so many ways. It's really, really cool to hear about everyone's different research with it, and it's obviously a ever-evolving field. But I do want to talk a little bit about you as well. So, you know, you just finished up with your thesis, you just defended it, and so what's next for you? Like, what are you moving on to after this?
Yeah, so I just defended at the beginning of March. Really exciting, and I will graduate in May. And the plan is to move to Gainesville and pursue a postdoc at University of Florida in Dan Hahn's lab. So it's really exciting, and you know, it's exciting to share that even in this challenging time for people to get positions in science. And, you know, when you start your PhD, you come in with an idea of potentially what you want to do, you know, what you want to do when you're older, when you're growing up. And so I came in knowing that I wanted to pursue a career in academia, and that has kind of stayed consistent throughout my PhD. I knew that I still wanted to pursue a career in academia. However, I've really grown to enjoy mentoring and teaching. And so I now want that to play a bigger part of my career in academia moving forward. And so I knew I wanted to pursue a postdoc with a mentor that not only, allowed me to develop new skills in the research lab, but also helped me develop professionally in that realm as well, with teaching, mentorship, or other roles with extension or things like that. And so I was really excited to meet Dan and pursue that option. And I am transitioning from my postdoc to a whole new research question, whole new set of bugs, and it's exciting, but it'll be a lot of work again. But again, I'm of the belief that you can be excited about a lot of different questions in science, and everything that you learn at each step can help and apply or help you gain skills to be able to answer questions in a different system or be excited about that other system.
Absolutely. And Dr. Hahn is awesome. He's super nice and like a big, huge name in entomology, and he has done a lot of really, really cool stuff. So do you know like what you might be working on here at the University of Florida?
Yeah, so Dan has a couple different sectors of his lab. Yes, he has a mosquito part of his lab. But the project that I am planning to work on when I come there is something involved with phytosanitary irradiation, which is basically a different pest management tool. The USDA is implementing an agricultural product that is, once it's been harvested, as opposed to just spraying it down with chemicals to kind of kill any bugs that are harvested with the product, can we expose it to different UV radiation wavelengths that will actually either kill and or sterilize the bugs that are on it? And so my project is really combining use of these different UV wavelengths to identify what wavelength is best used for that control, as well as in these different partial oxygen environments. So if we think about what our food is packaged in, it's packaged in these different plastic packages, I guess. And the environment that is then created with those packages is actually a lower oxygen environment than our atmosphere to preserve our food longer. And so we want to know, is this UV radiation effective not only if the agricultural product is exposed to our regular oxygen environment, but also if it's exposed to those packaging environments, as well as if the food is stored for long term in different cooler temperature environments. Can we change any of the USDA regulations with that? Can we make it so that there's more flexibility in the way that our food is packaged, stored, and brought to grocery stores and things like that? So I'm focusing in on two different pests, the sweet potato weevil and the corn earworm, and I'm excited to kind of see what these different environments do to the insects. So the USDA really only cares what environments kill the insects, what is the best use of the management strategy, but we will have some flexibility to determine, if these environments are not killing the insects, what are they actually doing to the biology and the physiology of the insect itself. And so that's kind of, I'm excited to see what kind of questions we can ask in that realm. Is it changing reproductive output? Is it changing lifespan? Is it changing the composition? Is it changing how they age? Is it changing, to bring about some of the different questions that I've already been interested in during my PhD.
That's super cool, and super applicable to the world around us. Everyone eats food, right? So it's obviously very applicable to everyone.
Yeah, I'm excited. I have this more basic sciences experience already, and so for my postdoc, I'll be getting more of an applied science set of experiments for experience. Yeah. And so I'm hoping then that, when I go on the academic job market here in a couple years, that really makes me a good candidate for not only just entomology departments, but general biology departments as well, showing that I can kind of ask these questions in different insect systems, depending on what resources are available at the university or the institution, and to kind of showcase how some of these basic biology questions may have application down the line. Because I really think that, it's important for scientists to understand the messaging of our work and understand kind of, the significance of what we're doing. Yes, we may be working on just a small project in the lab, but we need to know how to communicate the importance of that to everybody else to make sure that people realize that research matters.
Absolutely. That was actually my next question, was like, what's the big plan after for your postdoc, or do you know, or you want to go and be a researcher at a university, or go into industry, or what do you have any ideas for that afterwards?
So I still am planning to pursue a career in academia, something that balances both teaching and research opportunities and responsibilities. Maybe not necessarily at an R1 institution, maybe more an R2 or R3, something that I was familiar with my undergrad institution. And so I'm exploring some more, teaching opportunities and mentorship opportunities in my postdoc to kind of better prepare me for those experiences. We'll see. That's kind of the plan. I have really enjoyed teaching and mentoring more than I realized, and I enjoy seeing the excitement that the students have. So I not only have teaching and mentoring experience at the undergraduate level, but I mentored a high school student in the lab. I've gone and taught lessons at the middle school classrooms in Nashville. So I have really enjoyed seeing the excitement of the students when they are learning and figuring out, how these scientific phenomenon work and things like that.
Oh, that's awesome. I love to hear that. And that ties itself into the podcast too.
Yeah, exactly.
This is part of what you like to do. So awesome. So another question that I love to ask is what are some other activities or hobbies that you enjoy in, I'm sure the little spare time that you have, some of the things that you like to do to step away from work or from school. Yeah, what are some other hobbies that you enjoy outside of research?
Outside of research, yeah. Believe it or not, graduate students do have a life outside of lab, even though it might be small. But I really enjoy antiquing and thrifting around town.
Nice.
Really big into that. And then my husband is a musician in Nashville. So I spend a lot of time going to different music performances. So he's not your traditional country music artist, wannabe singer-songwriter. He's actually a classically trained woodwind specialist. So his main instrument is saxophone. But
nice.
So he teaches at Tennessee State University and at a high school in town. And so not only do I hear, classical saxophone concerts and classical woodwind concerts and things like that, but he also he plays clarinet in a Dixieland group around town. So
nice.
I hear that kind of music as well, and we go and support friends and things like that. So Nashville's been a really awesome opportunity to kind of feed that love of music and enjoyment of that kind of culture and everything like that.
Oh, that's so fun. Well, I think that's about it. Is there anything else that you have burning that you really want to tell us about, or anything that I might have missed, or anything you really wanted to tell the people?
I just really enjoy what you're doing with this podcast and your program.
Thank you.
I always think that it's a great, podcasts or other getting media is a great way to share the exciting things that we're doing in research labs in areas where you may think it's not that exciting in a lab, but really it is.
It is.
And I think this is a great opportunity to encourage other people to pursue careers in science, or just to, get a little bit more informed of what people are doing. Yeah. And so I really appreciate what you're doing with this. And I think it's awesome that you're gearing, this resource towards people that are at the beginning stages of their careers, and, people that are considering what to go into for research in college, or, they are really excited about something that they learned in a high school science class and they want to learn more about what's going on. So I think that's just really awesome.
Thank you very much. I appreciate that a lot. And I love doing it, and I love giving voices to researchers, because I think a lot of people just think of scientists as these reclusive, weirdos that do weird stuff that people don't understand in a lab, and that's really not the case. And I think you've really demonstrated that researchers are very personable and passionate about what they work on, and the stuff that they do really applies to everyone, and it's just a matter of getting it out there and making it into a medium that people understand and appreciate. And that's the whole point of my podcast, is to do that. So thank you so much for taking the time to do this with me, Jordyn. Thank you so much. This was super cool, and I love hearing about all the research that you're working on, and I'm excited to see what you do in the future, and maybe I might see you in the fall here on campus. So that's really exciting.
Yes, exactly. It's super exciting. Thank you so much for having me.
Of course.